great. Go ahead, David.
Great, thank you. Thank you for the introduction. Very happy to be here. Yeah, I'm glad to share my experience in helping out with research in spinocerebellar ataxia and kind of talk about what the disease involves and what research involves and future directions for research and things like that. So is it all right if I go ahead and share my screen?
Yeah, of course.
Great. All right. Can everybody see that all right? Oh.
It's still like loading.
Yeah, it looks like my Zoom's a little frozen. Okay, how's that?
Yeah, now we can see it.
Good, great. All right. Off to a great start. Love it. No. Yeah, just a little bit before I begin, a little bit of background about me. I went to Pitzer College in LA in California and graduated with a degree in neuroscience, and then was fortunate enough to get hired on as a research coordinator at the Geschwin Lab here at University of California, San Francisco. Do a bunch of different things in the lab. The lab does a bunch of research in SCA, which I'll be talking about, also MSA, Huntington's disease, MSA being multiple system atrophy, Huntington's disease, rapidly progressive dementias, things like that. But I mostly oversee the spinocerebellar ataxia studies, 2 clinical trials that we have ongoing and 2 observational studies. And it's kind of a burgeoning field for pharmaceutical companies. So those clinical trials are probably going to be increasing in numbers in the near future, which is exciting. But just a disclaimer, I'm not a physician, I'm not a PhD level researcher, but I do kind of run the research and the research visits on a day-to-day visit and deal with some of the operational things that go on in research. So I'm happy to talk about that. Don't know if it's going to get into the minutia of kind of more basic science stuff, but happy to provide any sort of insight that I might be able to. So I'll just move along here. So just a bit of a roadmap for what we're going to be talking about today. Briefly, I'm just going to talk about generally what is spinocerebellar ataxia. From now on, I'm just going to refer to it to SCA. So kind of saves us all a little bit of time here. Talk about kind of the genetic underpinning and what constitutes different types of SCA, how it manifests neurologically, how it presents with symptoms and disease progression, things like that. And then also kind of talk about what research we have ongoing here at UCSF, both observational and interventional things, and then future directions for research. So there's some exciting new trials on the horizon that I would love to talk about and that we're all very excited about. And then just a few things, kind of last words at the end as well. So what is spinocerebellar ataxia? I know you guys have done, NeuroHealth has done work on kind of ataxia and had people speak about ataxia in the past, but just as a reference, ataxia is a loss of muscular control and bodily movements. And I've kind of split it into 2 kind of categories here, one being spinocerebellar ataxia, one being everything else for the most part. But there are a couple different ways that you can get ataxia. You can inherit it through certain diseases like Friedreich's ataxia, which is a really common form of inherited ataxia. You can get it through other autosomal recessive kind of mechanisms for inheritance. You can also get it as like a secondary symptom from things like stroke, traumatic brain injury, alcoholism, which affects the cerebellum. So, can affect bodily function and gait and speech and things like that. They're also like mitochondrial ataxias, like X-linked ataxias, episodic ataxias, things like that. But for the purposes of this talk today, we'll be talking about SCAs, which are a group of progressive inherited ataxias inherited in an autosomal dominant fashion. So I don't know if people here have taken some baseline genetics, but luckily it's as easy as it gets in terms of like inheritance. So that works in our favor. Yeah. So just a refresher in case, you haven't gotten there yet. Autosomal dominant basically means that you need one mutated copy of the gene to develop the disorder, right? So there's a really clean little graphic here. You have an affected father who has one abnormal gene and one normal gene, and an unaffected mother who has two copies of normal gene. And then, you have 4 children, 50% of them are going to inherit, they're going to inherit a mutated gene. And so 50% of them are going to show the disease, so to speak. And, those are rough numbers, not always, doesn't always play out as cleanly as that, but that's how it works. That's how the inheritance works. There are usually people who have one copy of a normal gene and then one copy of an abnormal or mutated gene. However, there have been instances documented of people inheriting 2 copies of a mutated gene, which is really rare, but it has happened. Generally, you would think that since it's already such a rare disease, it would be some sort of like a child born out of the same family, and it just so happened that the disease ran in the family, and it just so happened that, perfect storm, you can have patients who are homozygous for SCAs, although it's super, super uncommon. Moving forward a little bit. So these are just a kind of snapshot of the different types of SCA. So on this chart here, we have 35 different types, but depending on where you look, they're up to like 45, 48 different types of SCA. Some of them, we just know that it's a genetically inherited ataxia, and we don't know what gene it is. We don't know, really much about symptoms and like age of onset and things like that. But we do know that it's an SCA. So that's why there can be up to like 40 something, and they're continuing to kind of characterize them and find more information about them. But if you just take a quick glance at this, you'll see this is the SCA type. This is the gene that's associated. This is the type of mutation. We'll get into that a little bit. And then there's some information about age of onset, average duration of disease burden of the different types, and then some of the symptoms and clinical features. We'll get into that, a little bit more, but just an overview. The SCA types are named in chronological order of like the discovery of them or their specific gene. So SCA1 is SCA1 because the ataxin 1 gene was the first one discovered, so on and so forth, But as you can see here, some of them, the gene is still not like assigned or not identified. So, it kind of depends on when they were discovered in order of the other SCAs. So I want to kind of narrow it in a little bit. And talk specifically about what are called polyglutamine or poly-Q SCAs. It's kind of a subset of SCAs, which depends on the type of mutation that causes the SCA. So when I say polyglutamine, it's the expansion of a glutamine encoding kind of CAG repeat section of the gene. So normal, like non-disease causing genes have a section of the gene that is a bunch of CAG repeats. Like everybody has that. In people with poly-Q SCAs, that expanded section or that repeated section is expanded. So there are even more repeats. So typically what you'll see is something like this table here, where for SCA1, that's the gene that's affected. Normally, 2 unaffected genes or 2 non-disease causing genes will have repeats in the range of like 6 to 35. And then from 36 to 38 means that like there's a possibility that they will develop symptoms, but it's not fully penetrant or not pathological. But when you hit this category, then it's pathological or fully penetrant, meaning that it's disease causing. And also it's worth kind of touching on the similarity between SCAs and Huntington's disease. And that's kind of part of the reason why our lab does both, is because Huntington's disease is also a polyglutamine disorder. And so, its genetic mechanism is dependent a little bit on those polyglutamine repeats. And that's kind of led to an adaptation of some Huntington's therapies and trials, especially kind of gene related ones to polyQ SCAs. And this table kind of has the polyQ SCAs here. The ones that are kind of most common, SCA3 is the most common SCA. It's also called Machado-Joseph's disease. You might hear it referred to in that way. Really interesting kind of origin story about that. Just briefly touch on it. It's there was actually a large cluster of SCA3 that originated in the Azores, the kind of Portuguese islands, and it was spread internationally, and it's really well characterized, and it's a really interesting kind of epidemiological case study of how we see genetic diseases spread and become like an international network of like an international disease network. So feel free to look it up if you want more information on it, but it's super interesting. And it's called Machado-Joseph's disease because the families of William Machado and Anton Joseph were like the ones that were kind of traced back and identified as sort of like some of the initial families that have contributed to a pretty large spread. You know, like there are other ways that SCA3 has come about. And it's not to say that they kind of spurred it on fully, but it's interesting that it's very well characterized, and there's an interesting origin story there, at least for some people with SCA3. As far as prevalence goes for SCA3, like about 2.7 in 100,000 have SCA3. That's a little bit hard to kind of, you know, compartmentalize as a number, but just for reference, Huntington's disease is about 1 in 10,000. So it's a pretty rare disease. Yeah. Let's see. Anything I want to mention on this anymore? Oh, one other thing is these polyQ SCAs, especially for some of the future trials that I'm going to touch on at the end of this. This is, these are kind of the targets for those. As far as pathological mutations, like it's often kind of a prerequisite for clinical trials because you really need somebody who's affected to be, you know, a good candidate for a therapy. All right. Also, just while we're on the topic of like repeat lengths and CAG repeats, it's worthwhile to mention that the CAG repeat length does have some sort of relevance towards disease characteristics and disease burden. So it's correlated with disease onset. Now, that's not like a hard and fast rule, but generally the larger the expansion, the earlier the development of symptoms and clinical characteristics. Though, you know, everybody's different, and we'll get to it later in the talk, how there can be a high kind of phenotypic variability between people with SCAs to some extent. Additionally, there's this kind of notion of anticipation, which is basically the idea that each offspring who inherits the disease occasionally has a greater CAG repeat length than the generation before it. And that increases with each generation. And so that can, you know, because expansion numbers can correlate with disease onset, you can get successively earlier and earlier onsets of the disease in the same family as it's passed down. Again, you know, these are just kind of like correlations. They're not predictive, but it has been kind of written up and documented. And one really interesting kind of way that this presents in SCA7 specifically is the anticipation can be so great that you could have a grandparent who hasn't developed symptoms, you know, maybe they're in their 60s, 70s, hasn't developed symptoms yet, pass it down without knowing to their child who hasn't developed symptoms yet, but maybe has a slightly earlier onset. Maybe they're in their 30s or 40s. And they passed it down to their kid who's now, you know, a newborn or like a young kid, and the kid starts developing symptoms before the grandparent does. And then they start tracing back. So that's just a really like exaggerated example of how anticipation could work. Doesn't usually happen like that, but in SCA7, it can happen like that. Okay. So I want to move on a little bit to talk about SCA as a neurological condition and a neurodegenerative condition at that. While there's like a bit of debate about the exact mechanism, and maybe people who do a little bit more basic science on this would have a better response about what they think the mechanism is than I would. But generally, these genetic mutations cause cell death mainly in the cerebellum due to things like transcriptional dysregulation, protein aggregation of kind of like toxic buildups of protein. Sometimes you'll see autophagy thrown around there, things like that, that basically lead to cell death and lead to, you never really want cell death. I mean, I guess in some instances you do, but not generally in the cerebellum, not neurons in the cerebellum. But yeah, in polyQ SCAs, these polyglutamine repeats create like extra protein, right? And those proteins just kind of build up and they form toxic aggregates. They can damage neurons and they lead to neuron death. So in spinocerebellar ataxia, as you may be gathered from the title, the cerebellum is primarily affected. And, the cerebellum generally oversees the coordination of movement related to motor skills, posture, balance, things like that. It can manifest in like speech, regulation of swallowing, things like that are a little bit less like voluntary per se. And additionally, like atrophy and cell death in the cerebellum can show up on an MRI, although it doesn't necessarily always. You can have somebody who presents clinically and like they obviously have SCA, but don't have much atrophy in their cerebellum. And it's not always super predictive, but genetic tests are. So, but in addition to the cerebellum, there are other structures that are affected. It kind of partially depends on the SCA type as well. But the spinal cord, brainstem, basal ganglia, pyramidal tracts, like can be involved. And have, ramifications and symptoms that result in that involvement. Additionally, I want to talk a little bit about, how does it present? How does it present clinically? What kind of symptoms do SCA patients have? And the kind of cerebellar symptoms are the most profound, like those are the ataxia symptoms that are kind of classic. And easily perceived by us. So this is stuff like dysmetria, like you can have like difficulty in like being specific with your movements. So if a neurologist does like a finger chase, kind of, my background's kind of cutting my fingers out here, but if you do a sort of finger chase, people will sometimes overshoot where you put your finger and then they'll settle back to like the midline and you'll go again and they'll overshoot and settle back to the midline. So it's a little bit of difficulty in regulating that voluntary movement and getting it specific and fine and precise. Also dysarthria, that's kind of, it's like classic ataxic speech. A lot of these symptoms are similar to as if somebody was drunk or something like that, because, alcohol can affect the cerebellum in similar ways. And so you can kind of get slurred speech, slowed speech, difficulty in being understood and articulating words and things like that. Additionally, you can have difficulty in eye movements. This can be a couple of different things. You can have overshoots in eye movements, just like with the fingers. You can have like, it kind of passes it and comes back, or undershoots. Also changes in saccadic movement. So like, pursuing something that's moving in front of you, generally you want it to be like a slow, steady progression, but sometimes it's jerky, and there are different ways that that's characterized in different SCAs that kind of have different forms of saccadic pursuit, or maybe not at all. Some have like nystagmus, things like that. As I talked about, a lot of people with SCAs have difficulty swallowing, hand-foot coordination, issues with gait. That's one of probably the most salient feature often, is it's a bit of a staggering gait, hard to kind of keep balance and maintain a sort of like, centered midline walk. A lot of people sometimes compensate by having a wider base. And additionally, sometimes you can have like urinary issues like retention, urgency, which like things like retention can lead to complications like UTIs and things like that. And then there are extracerebellar symptoms like dystonia, which is like kind of the fixing of the body in certain positions. Not as common, but some people do have it. And then also things that aren't quite as described in, like, you don't typically think of when you think of SCA, like issues with cognition, emotional regulation, things like that. And also there can be pain, like peripheral neuropathies that patients often have and sort of are surprised that they have, because it wasn't really brought up to them before that was possible in SCAs. And again, you know, I do want to just highlight the fact that I don't have the disease, and so I don't want to be speaking on behalf of people who do have the disease. But just want to kind of bring to light some of the symptoms that can be seen in these sort of disorders. Oh, and additionally here, if you're wondering about heart problems, that one, it's documented in Friedreich's ataxia. There's a mechanism that they've kind of studied and developed that links issues with cardiovascular, you know, issues with the actual ataxia. In SCA, it's a little bit less common and less known if there really is even a mechanism, or if it's kind of like spurious. But it can produce cardiovascular abnormalities, like in like abnormal heart rate variability and stuff like that. Yeah. Okay, so I just want to kind of break down the different types of SCAs a little bit, just in terms of how they present. Now, throughout all of this, you know, pretty much all SCAs have ataxia, right? And so that's something that presents often, and that's the most salient thing often for us viewing the patient, though that's not to say that that's the most important symptom for the patient, or they feel that, you know, that adequately describes their disease progression or their unique disease. There are a few characteristics that kind of differentiate different types of SCA. This one has like sort of more weakness of muscles, issues with vision, double vision, obviously ataxia, swallowing, spasticity, speech, peripheral neuropathy. Onset's like about 30 to 40 years old, although that is super variable as well. Disease progression and disease onset are really hard to predict, and that can also be a source of frustration for patients, because they're like, well, you told me that it wasn't gonna pop up until my 50s, but here we are. Like, so it's a difficult thing to accurately predict. In SCA2, you know, some eye involvement too. The actual retina is involved. There's some issues with regulating movements of the retina and things like that. And there's also atrophy, which instead of just like growing weak from lack of use, there's actual like degradation of the muscle. So it's a little bit more than just like lack of use. Also, you can have tremor, twitching, cognitive issues, and you know, as I said, the onset's pretty variable for that one as well. SCA3 is the most common one. Issues with sleep, fatigue, some emotional and executive functioning. Obviously, issues with gait and eye movement, speech, swallowing. There's atrophy in this one too. There's also like a lot of issues with fine motor control in the arms and in the legs. Also in the face, which is something that's interesting in SCA3. And a little bit, I suppose, of heart problems, although not as well documented. SCA6 is an interesting one. I threw it on because it's like a pure cerebellar SCA, which means that it's like its symptoms are really just like cerebellar symptoms, like balance, gait, swallowing, speech, things like that. But as I will get to, I guess I can talk about it now. It's interesting. It doesn't always predict it, right? Even though SCA6 is like the easiest one, boom, like open shut, pure cerebellar ataxia, there are people who have it, who have completely different symptoms and have a completely different onset. And they're like, well, you know, what am I supposed to do? This is frustrating me because it seems like this is the most open, like clear cut form of ataxia and the most predictable, but it just can have a lot of variability in how it presents from person to person. So And then just a few others here. SCA7 is worth mentioning because it has some retinal degradation, which can lead to blindness. And then in addition, all the ataxic symptoms. And then SCA10 can be, you can have some like epilepsy and issues with seizures in addition to the ataxic symptoms. So as I've alluded to, you know, all these SCA types have similar clinical presentations in like the ataxic symptoms and other cerebellar symptoms, but they can have some variability in kind of the more extracerebellar symptoms, and as well as, you know, disease onset, progression, and things like that. There's an example of, like, for example, one family who has SCA6, father had onset of symptoms in his 50s, like, you know, pure cerebellar, no issues with cognition, no pain, no peripheral neuropathy, nothing like that. Offspring got tested, had the same exact amount of repeats, developed symptoms in their early 30s, peripheral neuropathy, issues with cognition and emotional regulation, very little in the way of balance and cerebellar symptoms, at least initially. You know, that those might develop over time, but like things like that can happen even within families, and even within the same amount of CAG repeats, you can have variability in when and how the disease presents. Additionally, I just kind of want to touch on like cognitive considerations in spinocerebellar ataxia. So generally they're considered movement disorders, but there can be issues with cognition and memory and emotional regulation. There's a doctor here who does a lot of work in kind of cognition and how the cerebellum is involved in cognition and more executive function. His name is Dr. Jeremy Schmallman. If you want to look him up, feel free. Has some really interesting papers and interesting research. He developed this sort of like syndrome called the cerebellar cognitive affective syndrome, which kind of describes ways in which cerebellar deficits can lead to more executive deficits, like issues in executive function, linguistic processing, spatial cognition, and like emotional or affect regulation as well. Not everybody's going to have these complaints. And, you know, with a thing like cognitive issues, it could come from elsewhere potentially, but there possibility of SCAs to present with cognitive issues for sure. Just to give you a sense of like what he developed this CCAS scale, which is used in a lot of studies. It's used in our observational research as well. And so it kind of looks at like semantic fluency, like how many words can you name in a minute that start with a certain letter, or phonemic fluency. Well, I guess switch those two, but phonemic fluency, how many can you name in a minute that start with a certain letter. Semantic, how many of a certain category can you name, and then like category switching. So like one of the examples is like, I don't know, like a profession and then a job, and keep switching for a minute. And then kind of like recall of certain words, you give a list and then they're recalled later, and then reading a string of numbers from memory and then reading it backwards. And then this here is the cube draw. This is a good example of, firstly, they are just told to draw a three-dimensional cube. And this is an example of, you know, it's not like this is classic ataxia, but this is what you could see, how it might differ from somebody who had unaffected cognition. And then if the person taking the exam is not able to reproduce the cube, then you would do a cube copy, which they just have like a cube, you know, there in the space, and you just have to copy it. So just wanted to provide a little visual representation of kind of how that plays out. And then just a few other things, like what's similar about two items, and a little thing called go, no go, which it's not super essential that we get into, but it's just a little game of like, I tap once, you don't tap, I tap twice, you do tap type thing. Okay, and that is kind of a decent overview of kind of how different SCAs present. So I'm happy to answer any questions later about that, as I think maybe there will be some, but I'll move on to kind of what our research looks like at the moment at UCSF. So we currently have 2 clinical trials that we're kind of doing and that we're coordinating. Clinical trials also you'll sometimes hear them referred to as like investigational studies because they involve an investigational product. The first one is Biohaven 201 and the second one is Biohaven 206. So decent amount of similarities between the two, but we'll get there. So kind of context for Biohaven 201, and this is also kind of context for Biohaven 206. Basically, there's no FDA-approved medication for SCA. There are some drugs that physicians will prescribe patients to kind of mitigate symptoms, like gabapentin for pain. Some of the things for like this one here is also called Chantix. It's like a smoking cessation drug. Modulates like nicotinic receptors or has some activity there. It's thought to maybe help some ataxic symptoms. Also, a lot of people are on, well, not a lot, but a lot of the patients that we see, at least, are on baclofen or other kind of muscle relaxants. So you can get a lot of like rigidity and tightness and muscle cramping, especially in like SCA3. And sometimes people will prescribe riluzole, which is approved for ALS, FDA approved for ALS, and actually has some mechanisms that make it a good candidate for applications to SCA. So what this company Biohaven Pharmaceuticals did was they took riluzole, which is already approved, and they changed it a little bit. They made it a little bit safer, a little bit. They improved its pharmacokinetics and its bioavailability. So like, somebody could take it on an empty stomach and it would have the same absorption as like taking it with food and things like that. Just kind of made the drug a little bit easier to use. And started doing trials for it in SCAs, as well as like generalized anxiety disorder, Alzheimer's disorder, and OCD, OCD being obsessive compulsive disorder. And they call it troriluzole. So that's the clinical trial that we're currently working on. Both studies, Biohaven 201 and 206, both are looking at troriluzole. They're both sponsored by Biohaven. And just an overview for Biohaven 201. It was kind of an 8-week randomized control trial. So, double blind, nobody knew other than the program that dispensed medication, I guess, who was getting placebo, who was getting medication. And they were giving it for, qualifying subjects. So just as a sort of insight to how research works, there are, inclusion, exclusion criteria that people have to meet, especially for clinical trials where you're giving an experimental medication. You want to be very, very cautious about who you give that to, and you don't want, you don't want anything adverse to come of it. So there are a lot of kind of, nobody, you can't have uncontrolled medical issues. You can't be on certain medications. Like you have to have a certain threshold of functional capacity, things like that. And so they started this trial for troriluzole and SCAs. And the first subjects were enrolled in 2016. You may be asking why riluzole? Well, it has kind of like activity at the glutamate receptor, or it has neurotransmission. It modifies glutamate neurotransmission and interacts with like different types of ion channels and mechanisms that kind of facilitate in glutamate reuptake. So that's kind of a mechanism that's a bit disrupted in ataxias. So, they thought it seemed like a good candidate for application to SCAs. So this drug doesn't aim to stop or reverse symptoms. Rather, what it aims to do is to slow the progression. So, a year after taking the drug, are you in a better place than you were if you hadn't been taking the drug, which is a really hard thing for somebody with progressive neurodegenerative disease to like feel, right? Because it's easy for us as researchers to sit there and say, hey, your SARA score hasn't changed in a year and a half. And they're like, yeah, well, like, I don't know, it feels the same to me, right? But, it's difficult to design a drug, especially one that's not gene modifying, that does anything that's more than slowing the progression necessarily, although, maybe there are some that I'm not privy to. But the primary endpoint is the SARA score. This is the scale of assessment and rating of ataxia. I'll briefly show it. Don't want to run us too late here, but after the 8-week randomized portion, there's the option to roll into an open label phase where all the subjects could, if they qualified, be on medication, so there's no placebo. And that phase is actually still going. They're now in expanded extension week 190. Well, not in it yet, but it's currently renewed up to 192 weeks. So we still have subjects who were enrolled in 2016 who are still in the trial. Assessments are just kind of like questionnaires about symptoms, functional capacity, neurological exams, physical exams, suicidality scales. It's actually if it's not an FDA requirement, it's a strong urging of the FDA to track suicidality in any clinical trial that works, any neurological clinical trial. And also like safety data, like vitals, height, weight, blood work, urine sample, ECGs, things, par for the course in clinical trials. And also some measures of dexterity, cognitive assessments, and like any adverse events or medications that the subjects are on. This is the SARA. We don't have to go into it in too much depth, but it basically looks at like gait, stance, sitting, speech disturbance, finger chase. So like, neurologist holds a finger out here, other person holds a finger that mirrors it. And you see how much they kind of overshoot or undershoot within a certain window. Nose finger test, which is like, nose to out to the neurologist finger. That sometimes you can see like some endpoint tremor, which is like as they get farther to the extreme of the wingspan above the reach, there's a bit of a tremor that can kind of be visible. Also fast alternating hand movements. That's like on the thigh, you just keep going like this a lot. So it kind of tests that fine motor control. And then heel shin slide, laying down on an exam table, bring the opposite heel up to the knee and then drag it down your shin. And it's really looking at like, do you lose contact with the shin as you drag your heel down? So is there lack of fine motor control in the lower extremities to kind of, that prevents somebody with such a condition from, staying on the midline and touching their shin the whole time. Moving on to Biohaven 206, same sponsor, same drug as Biohaven 201. They changed the endpoint from the SARA to the F-SARA. So they developed a shortened, it's the functional SARA. They shortened it to only 4 items. And kind of their idea was to get rid of sort of the things that the SARA tracks that are not really that important to patients. Like, like a patient's like, okay, like, so what if I can't do the nose finger test exactly on, like I would rather have something that specifically targets like my ability to walk or my ability to speak, things like that. So they took out the eye movements, heel shin slide, finger chase, alternating hand movements, and kept gait, stance, sitting, and speech, and like kind of clarified the scoring a little bit too. So this study, they also expanded the randomization phase because 8 weeks was a little bit too short to see the full effect if there was one. And then they've extended it for 96 weeks afterwards of open label extension, which patients can opt into. Data's not out yet, it's still ongoing. Still have patients in the trial, but if the results are positive, that could be a big thing for drugs related to SCA, as there really aren't any at the moment. Now briefly, our observational studies. These are non-interventional studies that we have ongoing. So, no medication, just gathering data. First one is ReadySCA, which is the readiness for, readiness for clinical trials in SCA. I think that's something along those lines is the full acronym or the, expansion of the acronym. We are currently enrolling. So, did I throw that out there? But it's a longitudinal study for clinical trial readiness in SCA 1 and 3. And really what it's looking to do is gather like robust and comprehensive data on longitudinal changes in those 2 SCA types as the disease progresses. It was really looking primarily at early and pre-manifest subjects, so people who have really minimal symptoms, or have, positive genetic testing, or are at 50% at risk. So, their first relative has a definite diagnosis. They haven't been tested yet, so they're 50% at risk. They can enroll as well. We don't release the genetic test because there is genetic confirmation that we do as a result of being in the study. We don't release those, but there's a process for them to get that information. So it's a bit of an interesting kind of dynamic working with genetic conditions, and also working with patients who have a 1 in 2 chance of developing it, but don't know if they have yet, or if they have it, to begin with. But there are yearly visits for this. It includes kind of like motor testing, functional testing, physical neuro exam, blood sampling, questionnaires. There's also an optional lumbar puncture for cerebral spinal fluid collection, and an option for MRI sub-studies. So we don't do the MRI at UCSF, but there are different locations across the country that bring people in for MRI, and they see, okay, do we, over time, over 5 years, see any changes in their MRI? And can we correlate that with anything? Can it kind of predict anything? It's done internationally. So one of the aims of the study is to develop an international kind of the world's largest cohort of people with, those SCA types, and develop a really, really comprehensive data set for those. And also to get a good pool of good clinical trial participants as well. This is the other observational study. So this is CRC-SCA. This includes more SCA types. So 1, 2, 3, 6, 7, 8, and 10. It includes pre-symptomatic to symptomatic patients. There's no upper cutoff for how symptomatic. For pre-symptomatic, they do need genetic confirmation though. So we don't do 50% at-risk subjects in this one. Similar assessments to ReadySCA. In fact, a lot of people co-enroll in both because they're meant to kind of, you can cross off a bunch of assessments for both in one. So a lot of people do both. This one, one looks to develop a comprehensive data set, as well as kind of look at what changes in, or not even changes, what things in cerebral spinal fluid, what things in blood can be correlated with disease progression, what kind of genetic mechanisms can predict even disease onset and disease progression. So it's looking at, like ReadySCA is developing a comprehensive data set, but it's also looking at, okay, what can we, use as high confidence targets for future therapies and development of future therapies. Okay, so that's a little bit of the biomarker data analyzed, correlations between blood and spinal fluid and disease progression, genetic modifiers for onset and progression, also patient-reported symptoms, narrowing in on what are the most important and prevalent symptoms in the patient population, and how can we use that to inform future therapies and future developments. Specific biomarker data, they're looking at neurofilament light chains, which is kind of a measure of neuronal injury. There are models of disease progression and disease severity in HD based on plasma levels of NFL. So they're trying to do a similar thing in SCA and come up with, okay, can we look at the NFL levels in SCA, and can we, stratify it into certain categories of disease progression. Also poly-Q protein for those with poly-Q SCAs. They have the option to do a lumbar puncture, and they're looking at, okay, can we quantify the mutant protein, so the abnormal protein in the subject, and can we develop a kind of way to get an accurate readout of what that is, and a quantifiable measure of what that is, so that future protein lowering therapies, which I'll get to in just a second, have a way to kind of track their progress and make sure that they're actually lowering what they hope to be lowering. So last little bit here, future directions of SCA research. So I'll just kind of quickly touch on something called antisense oligonucleotides, which are generally referred to as ASOs, and also adeno-assisted virus vectors, which are AAVs. So ASOs, basically, the benefit here is they hold the potential for upstream mechanisms of therapeutic relief rather than just mitigating symptoms. So how they work, basically, it's a short single-stranded nucleotide that targets the specific piece of RNA that is kind of an issue in certain diseases, like in Huntington's disease or in poly-Q SCAs. And basically, it binds with that little piece of RNA, and there are a couple different ways that it can modify it, but it basically can reduce, restore, modify the mutant protein and the general protein expression. 2 ASO therapies have been approved by the FDA already, and there have been clinical trials that you may or may not have heard of that have been done in HD already, and now they're being kind of adapted for specifically poly-Q SCAs because of that similarity in genetic underpinning. Some of these trials, I would be remiss if I didn't mention, were stopped recently due to it was a little bit vague. They couldn't give too much away, but potential risk benefit profile, which doesn't mean that necessarily the risk was increased. It could mean that, it was risky for the participant, definitely, or it could just mean that there wasn't much benefit in it. But that's not to say that it isn't at least worth exploring in SCAs, because the science is solid, and studies have suggested that they do lower, the mutant protein that hopefully would be sort of the root of the disease and kind of assuage symptoms. Just a little graphic to visualize it. Because it kind of speaks for itself. But basically, the ASO would bind to the ataxin-3 pre-mRNA and degrade it. Therefore, less ataxin-3 protein would be produced, hopefully, decreasing any toxic aggregation of the protein. And sort of like, rather than dealing with the effects of toxic aggregates of ataxin protein with like medications, you would just decrease the levels. And so that would, mollify symptoms a little bit. This is not a gene therapy, as it only interacts with RNA and not DNA. But we have another direction of clinical trials and therapies, which is AAV. Basically, it's a protein shell that houses a small single-stranded DNA genome used to modify expression of target genes. And so this is a gene therapy, as it interacts directly with DNA. It's a naturally occurring virus. It's found in some places in our body, also just kind of in other places naturally. Kind of succinctly provides instructions that tell cells not to produce a specific protein. So theoretically, it could be used to block the expression of ataxin proteins in poly-Q SCAs. Studies have been done, just like the ASO studies have been done with this, with AAVs in HD. Currently, actually, my co-coordinator, Julia Glick, who may or may not be in the crowd out there, is running a trial for AAVs in HD, which involves kind of a direct neurosurgical administration of AAV, and aims to reduce the level of Huntington protein, so both the normal and the abnormal levels. And so that would it would work in a similar way theoretically for SCA3. And so it's kind of being pursued as a potential avenue for drugs. This is the general mechanism. I know it looks like something straight out of like a textbook, so I apologize if that gives you guys PTSD, but basically enters the cell, enters the nucleus, sheds or uncoats, its viral load, and then it integrates into the host genome, and then basically gives instructions to block the expression or the production of a disease-causing protein like Huntington or ataxin-3 or something like that. Hopefully, decreasing toxic protein aggregation and reducing, symptoms and disease burden. Other things I just wanted to mention, I think it's important, and the irony is not lost to me that I'm here speaking somewhat on behalf of people who are affected by this disease, and I don't want to speak on their behalf, but it's important to include patients and symptoms that are relevant to patients' quality of life, and involve them in kind of the discussion of, okay, how does a clinical trial work? What does it like aim to get? What do we want a drug to do? I think that's important. Not necessarily that, you know, that patients are designing the protocol, but at least it's important to get insight rather than just look at it as like a new avenue for the company to progress, you know, if you know what I mean. Additionally, there's, I think, just something about research in general is it's important, and there are obviously people who know more about this and who are doing this. So I don't want to come across like that, but you know, there's a fine line between having a trial that gets an exhaustive enough data set, but also isn't too exhaustive for the subject such that they, you know, aren't incentivized to come back in or not willing to come back in because it's just too taxing. And additionally, I just wanted to leave a few resources for patients and caregivers or people who want to learn a bit more. National Ataxia Foundation is a really good resource for like everything to like from education to like clinical trials, like cutting edge research, exercises for people with SCA, you know, recommendations for caregivers or family members of people with SCAs, and also like, you know, local support groups for caregivers and patients have from what I've been told, are really, really valuable. And also, you know, clinicaltrials.gov is the best place to go for upcoming trials and studies and stuff like that, because they have to be posted there. So, so it's a good way to kind of stay on top of that and see if there's anything in your area or anything interesting coming up. Yeah, other than that, I just wanted to include my contact information there. Please, anybody, feel free to send me an email. You can give me a call. No promise that I'll answer right away, but I'll probably call you back. Or if you want to mail me something, you could send it there as well. But thank you very much. I hope I didn't take too much of your time here. I really appreciate the opportunity to kind of talk about SCAs outside of work. It doesn't come up too often. So thank you guys. Happy to answer any questions.
Awesome. Thank you so much for giving that presentation. That was like super informative, and I personally learned a lot. So thank you. We do have quite a few questions in the chat, so I will just scroll up to the top here. So firstly, we had, how is the disorder affected if someone inherits both mutated chromosomes?
Yeah, that's a good question. I wish I had a better response to that. I don't know, because it's so rare. I don't know that there's enough data to really generalize how that presents. Yeah, I wish I could give you a better answer, but I don't think it's necessarily correlated with a worse disease burden, but that might just be because there's not that much data on it.
Right, right. Okay, thank you. Yeah, and then there's, what is the prevalence of SCA3 in the overall population?
Yeah, good question. Let me see. I have the number here. It's 2.7 in. I'm just gonna scroll up a little bit. 2.7 in 100,000, which is a little bit like I kind of touched on, is a little bit hard to wrap your head around, but that's 2.7 in 100,000 of SCA3. For Huntington's disease, which is, you know, a more prevalent disease, that's 1 in 10,000. So, you know, do with that what you will.
Right, okay, thanks. And next is, if you have a large polyQ CAG repeat, can you still be non-diseased?
Not if you are past the kind of cutoff. So I can quickly back up to the slide on cutoffs for penetrance. So here, if you have a really large CAG repeat or polyQ, you know, CAG repeat, then, and it's like really large, then you would be in the pathological section, and like pathological means that it would be disease causing. Now, it doesn't predict necessarily, like it doesn't 100% predict what your disease is going to look like and what the progression is going to look like and when it's going to develop. Although, like I said, you know, there's some data that correlates progression or disease onset with the length of the expansion. But that's not necessarily a hard and fast rule, and it's not necessarily true for everybody.
Okay, makes sense. All right, and can gene therapy be used as a treatment for SCA?
Hopefully. We don't know yet, but that's kind of the aims of that AAV technology, and those trials are kind of to see can we attack SCA from a gene therapy, a gene modifying direction.
Right, okay, cool. And do you know if SCA is screened during carrier screenings before pregnancy?
It's a good question. I think it's rare enough that it isn't, but I also don't quote me on that because I'm not positive. I know that some people do, IVF, in vitro fertilization, to select the ones that don't carry the mutation, which isn't exactly answering that question, but it's a way that some people get around that issue of, having a child with a known mutation. How do you ensure that the patient child doesn't have the mutation?
Right. Yeah, that was an interesting question. Thanks, Sheldon. And then I actually had a follow up to this next question because I had a question about this myself. But the question was, do we know why there's variability in disease onset? And I was wondering, like, I've never uh, like personally heard of a neurological disorder that like the next generation has like an earlier and earlier onset. Is that like unique to SCA, or is that something that you have seen in like other neurological disorders as well?
Good. That is a good question. Let me just, yeah. I I I I want to say that it's, seen in HD. Um, yeah, it is seen. Yeah, it happens in HD as well.
Okay,
Yeah.
that's cool. Yeah. So do you know why there is a variability in the disease onset, or I guess like that's just like has to do with the genetic makeup?
Um, well, I mean, part of the best answer that at least like the scientific consensus from what I've heard and seen is just that there's a correlation between length of repeats and age of onset. But again, there are exceptions. And as for the actual mechanism of why that necessarily happens, I mean, maybe it has to do with having more repeats means more toxic aggregates means a more aggressive disease progression. But there's a lot we don't know specifically about when the disease presents, like how the disease presents and why it's different between people, between families, or sorry, between families, within families, and between people who have the same exact amount of repeats.
Right. Okay.
Yeah.
Yeah. No, that's super interesting. It looks like I missed one. I feel like you kind of just covered this a bit, but do you know why the CAG repeats can cause SCA?
Yeah, that one's a little bit more like solid in its explanation. Generally, people think that having those extra repeats means that extra protein is produced, and when extra protein is produced, it can form toxic, aggregates and conglomerations of protein that kind of really get in the way of neurotransmission of cellular function and can lead to like cell death and the symptoms that you see in SCAs.
Okay. Okay. Yeah. Thank you. And last one, could ASO be used as a vaccine?
Do you mean like preventatively or I don't know if we have the ability to clarify questions here, but I'm not sure. I don't think it would be used like preventatively, if that's what you mean, but it could be used in the sense that somebody would get the therapy when they're really early on in their disease process, or even before they develop any symptoms, maybe to kind of like get it before it kind of blooms. I don't know if that really answers your question about whether it can be used as a vaccine or not. I think it would be used more as like a therapy, to get at the root cause of the disease.
Right. Sheldon, was that like what you were asking? Like, did that answer your question?
Yeah, so my reasoning was because it's used for like the protein that causes the disease, I think, right? So
Mhm.
then I was thinking like if someone could be pre-screened and it kind of shows that they have like those repeats, if they can like get kind of like the ASO beforehand before they have symptoms, and then will that be a preventative measure for it?
Yeah, definitely. Good question. That's actually one of the main criteria for this for upcoming ASOs is you really want the patients to be like pretty either pre-symptomatic, like you said, they have a known family member, there's a known genetic mutation in the family, and they've been tested when they were really young because of it, but they don't have symptoms yet. They're a good candidate for these sort of things to kind of get out in front of it. Also, people who are early symptomatic as well um, tend to be the patients that these studies are hoping to enroll.
Okay, yeah, great. Is there any other questions that they didn't get a chance to write in the chat or that they just like thought of now?
Yeah, can you guys hear me?
Yeah.
Okay, great. So this isn't quite related to the research topic that was discussed today, but as a student in the Bay, I'm a high schooler, so I'm not that familiar with how research is structured and all that. But this presentation gave a lot of insight into how interesting it is and how it's conducted. I was wondering if UCSF holds seminars or some sort of like talks open to the general public regarding certain research that may be interesting to students that aren't enrolled. I
Yeah.
wanted to ask about that.
Yeah, definitely. I know they used to. So I started a year and something ago. And so I've been here in the pandemic the whole time. I know they used to have guest speakers on campus all the time. And I'm pretty sure that some of them were open to the public. I don't think all of them were, but I think a lot of them were. I'm pretty sure there are a lot of public seminars that they have that are now virtual. If you maybe want to send me an email, I can look into it and send you any information that I can find, because I don't necessarily know off the top of my head, but I'm sure that there are opportunities for that.
That'd be great. Thank you so much. I'll send over.
Yeah, of course.
Awesome, thank you. That's great. I'd actually be interested in that as well, so thanks for asking that. Any other questions that anyone wants to finish up with? No? Okay, I'm gonna go ahead and stop the recording.